All right, it's my pleasure to introduce Sujita Balami. She's a PhD student with the Mississippi State University's College of Veterinary Medicine, working with Dr. Matt Griffin at the Thad Cochran National Warm Water Aquaculture Center in Stoneville, Mississippi. Her research focuses on bacterial pathogens of farm-raised catfish, particularly the development of vaccines against Edwardsiella piscicida in channel and channel blue hybrid catfish. Looking forward to your talk.
Thank you, Dr. Fells, for the introduction, and thank you, AFS-FHS, for giving me this opportunity to present. Today, I'll be presenting a part of my graduate research on evaluating oral and immersion delivery of an Edwardsiella piscicida vaccine in hybrid catfish. Shout out to all my co-authors who have helped me throughout this project. And I would like to give some introduction and background information. Ichlud catfish are one of the most important farm-raised fish species in the United States and contribute significantly to the U.S. economy. It was predominantly occupied by channel catfish culture before, but hybrid catfish, which is a cross of male blue and female channel catfish, is increasing in popularity because of its high growth rate and production, as well as increased disease resistance to several channel catfish pathogens. Right now, the industry is occupied by 60% approximately by hybrid catfish, and this shift has coincided with increased E. piscicida associated edwardiellosis, which is caused by Edwardsiella piscicida, and it infects wide range of fish species and affects fresh brackies and marine fish, as well as attributes to significant economic losses in the US as well as the global aquaculture. One of the problems of this pathogen is its high genetic diversity. Till date, there are 5 genetic variants that have been reported only in catfish, and it creates problem in the fish management as well as vaccine development, because one vaccine may not work for all the genetic variants. According to the CVM Aquatic Research and Diagnostic Laboratory Report 2023, we can see that in all the total E. piscicida case submitted at this laboratory, approximately 90% of this diagnosis are from hybrids, and suggests it's a huge problem. Despite this, there are no commercially available vaccines for E. piscicida in the US. So one of the thing we can do for creating a live attenuated vaccine is the bacterial attenuation, which can be done through random mutagenesis or targeted mutagenesis. Targeted mutagenesis is highly efficient but carries with it some regulatory hurdles and challenges with public perceptions of GMOs, whereas random mutagenesis is easier to perform, and we have 2 methods of repeated passages in extreme growth conditions and serial passaging in increased concentrations of antibiotics. One of the antibiotics that are used for creating these mutants through passaging is rifampicin, which are known to cause series of mutations in beta subunit of RNA polymerase gene, which makes the bacteria avirulent or less virulent compared to their wild types. And we are trying to use this method because there are several FDA-approved vaccines that were created this way. For example, Edwardsiella ictalurae immersion vaccine, Fibrobacterium columnar immersion vaccine, which are both commercialized, and Edwardsiella ictalurae vaccine, which is an oral vaccine that is being given under veterinary prescription right now and is effective. So with all this background information, we hypothesize that E. piscicida can be attenuated using the rifampicin passage and used as a potential vaccine. And the objectives of my study are to develop rifampicin pass Edwardsiella piscicida, test the safety of these mutants, and to evaluate the efficacy of the mutants via oral and immersion vaccination in hybrid catfish. Moving on to the methods, we first passes the isolate in the rifampicin media initially starting at 0.5 microgram per ml and then passed it on to higher concentration with increments of 5, 10, and 15 microgram per ml. We expanded them in broth, and cryostocks were made at each step. This is a visual representation of how the mutants were made, and we stopped the passaging at 360 microgram per ml, creating 17 mutants from this process. Previous experiment in our lab, we tested the attenuation of these 17 mutants with intracellular injection challenges, and the results from previous experiments reported that we have 3 isolates that were attenuated and protective with relative percent survival of greater than 75%. And we decided on going forward with these 3 mutants for our oral and immersion vaccination. We also included one more isolate which was developed previously at NWAC, which showed similar results. For the oral and immersion vaccination, we set up the experiment with 20 fish per tank and 6 replicates. Immersion vaccination was done for 1 hour at dose of 1 times 7 CFU per ml with a single dose of vaccination. For oral vaccination, we coated a pound of feed with 100 ml of diluted culture and fed the fish with that coated feed at 4 times 7 CFU per gram of feed for a single dose. The fish were monitored for 42 days post-vaccination. And we did not found any mortalities in any of these vaccinated fish for this time period. Bacterial persistence was checked by sampling the fish at 1, 2, and 42 days post-vaccination. We did kidney cultures from the sampled fish, and the colony that grew were used for real-time PCR. At 42 days post-challenge, at 42 days post-vaccination, we used Idwasella piscicida strain S11285, which is found the most virulent here at NWAC for challenging the fish with intracellular injection at the dose of 4 times 6 CFU per fish. And dead and moribund fish were used for kidney cultures for infection confirmation. We also collected blood at 42 days post-vaccination, harvested the serum, and checked the anti-E. piscicida IgM activity using indirect ELISA. Now, moving on to the results. We isolated all the candidates from orally vaccinated fish at 1 day post-vaccination, while only 2 candidates were recovered from immersion vaccinated fish at this time point. Whereas at 2 days post-vaccination, 3 candidates were recovered from the kidneys of orally vaccinated fish, while only 1 candidate was isolated from immersion vaccinated fish at this time point, indicating that the vaccine isolates were entering the fish, causing the infection, but not causing the disease. But at 42 days post-vaccination, no bacteria were isolated from kidneys, indicating the clearance of all vaccine isolates at this time point. Looking at the survival results, unfortunately, we hit a very high challenge dose, and more than 80% of the fish in our challenge died. In hindsight, we should have targeted a lower dose, but we wanted to investigate the effectiveness of the vaccine against an extreme dose. So, however, because this is an injection challenge, we believe that we have overwhelmed the protective effect of the vaccine. And also, I want to indicate that in our previous challenges, we used to have our peak mortality only after 2 to 3 days post-vaccination. But in this experiment, we had our peak mortality at 12-hour post-vaccination, which really indicates that we hit them very hard. And as Matt said, we smoked them, so we did not see any effect. However, in one of the vaccinated group S11285R, in the immersion vaccine group, we saw numerically higher survival. And for the oral vaccine group, we found SO7346, which is numerically higher in survival. But I would like to point out that they were not statistically significant. For analyzing the ELISA data, we thought of 2 analyses. One is endpoint titer method, in which we select a cutoff point of observance, and we compare the endpoint titer of the samples with each other for statistical analysis. Whereas in the midpoint titer, we use these observance values to create a sigmoidal curve and determine the midpoint titer uh, here at C, and then compare that midpoint titer of the samples with each other. But when we do our endpoint titer method, as you can see here for the vaccinated and non-vaccinated group, you see the difference is very low, because this only gives a specific values that is determined at that point and does not account for the plate to plate variations and does not give the more accurate representation. But if we do a midpoint titer, as you can see in the orange dotted lines, we can see a clear difference between the vaccinated and non-vaccinated group. So we decided on using the sigmoidal curve for our analysis, but unfortunately, we did not get the appropriate shape of sigmoidal curve because at the dilution which we were performing our ELISAs, as you can see in each of these treatments, we were a little bit on the lower side of the curve, not on the upper side, which was important for our analysis. So fitting into the sigmoidal curve was not possible because our data were only located at the tails. And also, we did not have enough serum samples for our experiment for lower dilutions such as 1 to 2 and 1 to 5. And also, I want to mention that we do not have a very strong anti-E-Picida antibody response in hybrids compared to anti-E. which has been done here before at NWAC. So this might be the reason that we are not seeing that difference between the treatments. For looking at this chart, we decided on showing the difference between the dilutions at 10, 20, and 40 for immersion vaccination and oral vaccination. You can see that at 1 to 10, there are significant differences between the treatments and the controls. But when we go at higher dilutions, 20 and 40, these are masked. So there is an influence of choosing the endpoint titer and the statistical analysis and also the results. Now, moving on to the conclusion, we saw two isolates, SO7346 mutant and S11285, had greater survival compared to other treatments, and anti-E-Picida antibody activity in these treatments were numerically elevated. So these data suggest that these isolates have potential as oral and immersive vaccines for hybrid catfish against E-Picida. However, further investigations for efficacy is warranted at less extreme challenge doses, and serological analysis should be repeated with more numbers of large size fish where we can do our dilutions at 1 to 2 or 1 to 5. So with this, I would like to thank my funding agency, Mississippi State University and FFAR, my all collaborators, as well as my committee members for all the help, and I'll take any questions.
Great presentation. Thank you. Folks, we have time for a very quick question, or you can drop them into the chat. So we got a question here in the chat. Sujita, was the attenuation by the rifampicin resistance stable over multiple in vitro passages or after recovery from the fish? And did you assess any potential reversion to virulence?
Thank you for the question, but we have not checked the multiple passaging yet. We were deciding on doing it after we finish our vaccine trials, and also we had some troubles with wet lab last year, so we could not do it. So that's our next step. We will be doing the multiple passages and looking at if the mutants revert to virulence.
Great. Always more work to do. Getting lots of compliments in the chat on the job well done. Thank you.
Thank you.